project is to design a new molecule, a new drug, and our project name is From Molecule to Medicine. From Molecule to Medicine, drug design and development. All of these different compartments represent each department. Okay, in all these departments represent specific research area. Okay, so whenever we conduct any research to find or to develop or to design new drug, so first of all we choose a disease. Okay, choose a problem. So what is the problem in our biological system? Let's suppose we take the example of arthritis. So arthritis is a disease which causes inflammation. So how to tackle inflammation, how to overcome inflammation, how to subside inflammation. Therefore, first of all, we identify the target. Then what is the main target? Which is the specific area? Which area is to be selected? So then, for example, let's suppose this target is protein. This target is like any enzyme. This target is, let's suppose, to be any receptor or any like gene abnormality. So we have a lot of different types of drugs which are already reported, which have anti-inflammatory activity. Okay, so we have all these 1, 2, 3, 4, 5, all these different compartments. In each of all these different compartments are responsible to evaluate this anti-inflammatory activity step by step. Okay, so in this area, all these molecules which are used here, they would be considered as the therapeutic molecules, because these molecules have some therapeutic properties. Let's suppose we start from phytochemistry. Phytochemistry is the plant chemistry. All those active compounds which are collected, which are isolated from plants, and which are responsible for anti-inflammatory activity, which have the analgesic activity. So all that such compounds which are already evaluated, which are already subjected to anti-inflammatory activity, and their anti-inflammatory activity is reported. So such compounds will again be evaluated, and their structure can be elucidated by through analytical lab, and then they will be subjected for further in vitro and in vivo activity. Similarly, we have some structure analogs. The structure analogs are all existing drugs which are responsible for anti-inflammatory activity, plus all those compounds which are having the structural resemblance to already the existing drugs. Let's suppose all these existing drugs, some commonality in their structure. Okay, first of all, let's suppose this is the aromatic area, and all these reported drugs having the aromatic area, and all these compounds having some specific functionality like carboxylic, the acidic area. This acidic area and the aromatic area, some polar and non-polar area, is responsible to subside the anti-inflammatory activity inside our biological system. Okay, so all these such compounds will be considered as a structure analog. So such structure analogs will be first of all synthesized, then their structure will be validated, and then they will be evaluated for in vitro and in vivo activity. Similarly, we have another compartment which represents another department which is called virtual screening. Virtual screening is actually the computational study. All those proteins or receptors through software which their structures are designed in a software, and through software all these ligands, all these structures, they are interact with the receptor in 3-dimensional program, the computer-based program. They are dog means that they will be interact with the receptors, and their matching will be studied, that how much this ligand or this structure is matched with the pain receptor. So all such compounds first of all will be synthesized, they will be docked, and they will be like analyzed, and then they will be evaluated for further activity. Similarly, this is the much more important area. This is called the fragment-based drug design. That small fragments will be chosen. They will be individually entered with the receptor. For example, let's suppose this is the receptor size. Look at this area. That this is the receptor area. This green one is the structure or the portion or the fragment which is already fit, and then this the red one or the purple one which is the second fragment. It will be also fit in another area of the receptor. Similarly, the third portion will also be adjusted inside this receptor. So all these fragments which are adjusted inside this receptor will be combined, and they will make like a new therapeutic molecule. Okay, this is the way that how fragment-based drug design through we will make a new therapeutic molecule. Similarly, we have the combinatorial chemistry. This is also called HTS, a high throughput screening, through which we have a lot of different bunch of chemical compounds which are thoroughly studied. They are synthesized, they are elucidated. You know, this area, this is the elucidated area where we have all different type of analytical instruments like UV spectrophotometer, we have infrared spectroscopy, we have proton NMR spectroscopy, CFT NMR, tape, different type of other analytical techniques, and then all these such compounds, whenever they are evaluated for further in vitro activity. So all such compounds which are analyzed or whenever they are synthesized, they will be subjected into in vitro activity. Okay, so during in vitro activity, they will be evaluated against living tissues. Like for example, let's suppose we have any cancer tissue. So they will be like whenever we perform any bioassay, okay, the tissue culture. So all such type of activities will be evaluated here as well. Microorganism, we also be study will also be run here. Okay, antimicrobial activity, antifungal activity. Okay, so all these such activity will be performed here. Okay, so whenever molecules are passes from in vitro activity in they will be further subjected into in vivo activity. So all these therapeutic molecules which are subjected into in vitro activity, so that would be considered as the lead compounds. They will be called lead compounds. Okay, so lead compounds, whenever they are treated against this in vitro activity, they will be further promoted into in vivo activity. In vivo activity, all these such compounds will be further evaluated for their biological activity, okay, against living organisms, like for example mice, rats, frogs, rabbits, guinea pig, dogs. Okay, so all such type of animals which are used for this is pharmacological studies. Okay, and their behavior, their different tissue, their blood, their different biological system, everything will be evaluated here. Okay, the homotoxic components are also supposed to be generated or metabolites are generated. Okay, so such lead compound will be evaluated here, and they will be called. And so this is the lead optimization area. This is called lead optimization area. So whenever all such compounds are screened from the passes through the lead optimization area, so few of the compounds will be selected here. Let's suppose, for example, 6, 7, 8, 3, 4, 5, depend upon the different biological system or the category of the disease. So these compounds which are further evaluated for next human trial, because this trial will now be called the clinical trial, because all these animal study is called the pre-clinical trial, and now they will be called the clinical trial. Okay, so such compounds which are passes from clinical study to the human study, they will be called the candidate drugs. Okay, so in candidate drugs, we will took from 20 to 30, 40, 50 individuals. They will be healthy volunteers. They will be selected, and this area study will be continued from 1 to 3. Okay, so also toxicity or pharmacokinetic study, everything will be evaluated here, and then it will be further promoted into clinical trial phase 2. So all the candidate drugs which are passes successfully from clinical trial phase 1 to clinical trial phase 2, whenever they are promoted, so it means they are comparatively much more safe. They are comparatively much more safe. Okay, so this study will be continued from 3 years to 6 years. Okay, and here we will took like healthy volunteers as well as the patients. Okay, and the number of volunteers will be from 100 to 300. 100 to 300 volunteers. Okay, so whenever this study is successfully run, then all such candidate drugs, whenever they are further evolved, okay, and there is, for example, there is no such abnormality or there is no such type of any like such toxicity is reported, so they will be evolved for the further phase 3 trial. This trial is the in this trial the number of volunteers, number of patients will be from 1,000 to 3,000. Okay, 1,000 to 3,000. Okay, so in this area, from the clinical trial phase 2, those will be calculated. Okay, and this will be further evaluated. Everything will be further evaluated. Either they are pharmacokinetic study, they are dosage form, their safety, every protocol will be evaluated here. Okay, and whenever, for example, let's suppose among all these candidate drugs, whenever 1, 2, or 3 compounds are successfully passes through all these different compartments or different trials, so they all will be registered through Food and Drug Administration. So whenever they are FDA approved, then further a large scale volunteers will be research will be further subjected. Okay, and there will be a large scale manufacturing, and this study will be continued from 6, 7 years depending upon the severity of the disease, severity of the area, the problem. Okay, and then after whenever it is passed from this area, then we will manufacture such compound for a large scale, and this is called the pharmaceutical industry. And after pharmaceutical industry, whenever they are commercially synthesized and prepared, and so then after that there will be the pharmacovigilance, and this study will also be continued for a long run. Okay, so this is all about the drug design and discovery and development. Thank you.